Good evening, everyone. Thank you for making the time to join us today. This is Perspectives by Kivani Initiative. Today's webinar is about performing virucidal efficacy studies under GLP. The onset of COVID-19 pandemic has seen a surge of products claiming virucidal and antiviral properties, reducing the prevailing infection burden. And any antiviral intended to protect public health must be registered with the National Regulatory Agency. As part of the process, efficacy data under good laboratory practices must be generated. And that is what will be talked about by Dr. Rahul Fadnis. Dr. Rahul Fadnis pioneered setting up a first GLP certified virus clearance and testing service lab in India. And he has several publications in peer-reviewed journals and patent to his name. And Rahul is an experienced biotech professional in viral safety, clearance, and vaccine development. Currently, Rahul heads the viral testing facility at Syngene International Limited. Before I hand over the reins to Rahul, just a little housekeeping. If you have any questions during the presentation, please type them in the Q&A box that is available in your Zoom control panel. And once the presentation is over, we will bring up your questions during the dedicated Q&A session. Time permitting, we will answer all the questions. In the event we are not able to, please don't worry about it. We will publish the unanswered questions on our website. We will also share a copy of the presentation which Rahul is going to use today and a recording of this webinar on our website, which is kivani.in. So now, without further ado, let's turn the time over to Rahul. So Rahul, welcome, and thank you for sharing about the viral efficacy studies under GLP.
Thank you, Vijay. Good morning, good afternoon, and good evening to everyone in your respective time zones. It's a privilege for me to be here and presenting in the webinar series, which is being broadcasted by and organized by Kivani. And today I'm here to talk about performing the virucidal efficacy studies under GLP and the kind of experiences that I have come across while conducting those studies is what I'm going to share about. So before we start on with this webinar, just a few things that already Vijay has talked about the housekeeping rules. And then today in the webinar, I'm going to talk about what are virucides, their basic properties, how virucides, studies for virucides are conducted to evaluate their virucidal efficacy, and the process flow of conducting these studies under the GLP conditions. And at Syngene, how do we perform such studies so as to ensure that you get a quality data out of these studies. And I'm going to present this in form of some case studies as well. And in the end, we'll be talking about some key takeaways. And of course, I will keep the floor open for Q&A session at the end of this presentation. So what on virucides, before I start on, as we all know, we are going through this tough times of COVID-19, and recently we have celebrated the anniversary of COVID-19 as well. So so far the experience tells us that we should follow an SMS rule, which stands for social distancing. Keep a safe distance of close to 6 feet when we are talking to each other. Second, M stands for masks. So please wear mask as and when you are stepping outside your homes or moving into your office spaces and having discussions with your colleagues. And the last S stands for use of sanitizers or soap solutions as and when they are suitable. So keep sanitizing yourself. And with that, I'm going to touch upon what is the current topic using the disinfectants as the sanitizers are one of the disinfectants that are being now more prominently being talked about and used in our day-to-day lives. It has become a part of our day-to-day routine. So how these virucidal disinfectants are tested and evaluated for their virucidal efficacy is what I'm going to talk about. And how these studies bring about confidence for us so that we can use them and feel safe. So the disinfectant market globally is quite a big market, and the research market research studies tells that it stands currently at around $3.1 billion and is growing at a compounded annual growth rate of 3%, and it's going to touch $3.6 billion by the end of 2025. So some of the key factors that are driving this market is increasing prevalence of hospital acquired infections. Second is prevalence of stringent regulations for the use of the surface disinfectants. And of course, now with the surge of, with the requirements of disinfectants becoming more and more prevalent, the demand of the disinfectants is drastically going up. So coming back to the basics, what are virucides? The definition says virucides are physical or chemical agents that deactivates or destroys the virus. So they completely kill the virus off so that it makes their effect and it completely neutralizes their effect. So, and we need to understand this is really different from the antivirals. So the antivirals typically prohibit the growth of viruses, whereas the disinfectants completely deactivate or kills the virus. So that's how they are called as virucides. And these are not intended for use inside the body. And most disinfectants that are not intended for use on the surface of the body as well. So some of them are not fit for using onto the humans, but they are to be used on the surfaces so as to make the surface free of viruses. The virucides are usually, they come with their own set of instructions for safe use and how effective they can be. So all the instructions should be followed in order to ensure that the disinfectants are effective in their use. Some of the examples of disinfectants are like soap and water, which is a very common form. Second is isopropyl alcohol, which is effective for some form of viruses, but not all. Ethanol, bleach is one of the very commonly used disinfectants in our day-to-day households also. And my slide also tells about some of the commonly used disinfectants that are prevalent in the market. To name a few are like Vivcons, Lysol, interferons, and some of the virucides have their applications in the veterinary industry, in the human industries, as well as in agriculture. So these virucidal products have susceptibility to the different forms of viruses and different. They also they have their mode of action on different structures of the viruses, and that's how they are effective. Some of them are effective on the nucleic acids. Some of them are effective on the proteins, the envelope proteins. And these are the ways in which the virucidal products have their effects. So in general, the morphology of the viruses typically falls in two categories: envelope viruses and the non-envelope viruses. The basic difference between the envelope and the non-envelope viruses is that the non-envelope virus has the protein coat on its surface, and that's the one which makes it more resistant. And the other category, which is the envelope viruses, are the ones which have envelope coat on its surface, which is the protein, lipid bilayer membranes, And this makes this virus, which is makes it more labile. As a result, the effect of virucides tends to be much more prominent on some of the enveloped viruses than the non-enveloped viruses, and hence we need to keep in mind that all the virucides will have to be potentially tested against both the enveloped and non-enveloped viruses to be ensured that they are effective against all forms of viruses. Otherwise, they can also be categorized into their effects based on their effects on enveloped versus non-enveloped viruses. So I'm giving some of the examples of the mechanism of actions for the type of chemicals that use as disinfectants, like aldehydes, which bind to the protein, and that's how makes the structure of protein makes it into an inactive forms. It interacts with proteins or DNA and RNA. Some halogens, they penetrate the membrane and oxidize the proteins, and that's how make them inactive. Peroxides penetrate the membrane. Phenolics also poison the protoplasm and damage the cellular membrane, and in turn they also work on the envelope viruses. Quaternary ammonium compounds damage the cellular membrane and disrupt the membranes. Bivinates damage the cellular membrane and coagulate the intercellular constituents, whereas some disinfectants, which work basis of their pH, they denature the protein surface, and that's how they are more effective on the envelope viruses than the non-envelope viruses. So typically talking about the characteristics of the disinfectants that one would like to have, is that it should have a fast action, it should be durable, it should be non-toxic, impervious to environmental factors. Then it should have a broad spectrum for its biocidal activity, which should ideally cover bacteria, viruses, and fungi, and should have precise determination of the scope of disinfectant action, so that it facilitates the correct disinfection method application. So that the understanding is correct, and that's how it can be, the directions of use can be rightly prescribed. So to test these disinfectants for their biocidal efficacies, there are different guidelines that are available. Some of them I am listing here. They are, of course, they are not the complete list, but just for the guidance of the viewers, they fall under different categories, and it has to be tested based on what is the mode of application and where they find their applications in which industry, and that's how it has to be decided. The relevant guidelines should be used for testing the virucidal products. So with this, I now come to how one should plan to conduct these studies, so that you have the right objective being addressed when you are testing the disinfectants. So as I said in my early part of the talk, that the viruses fall into two categories. They are envelope viruses and the non-envelope viruses. So the envelope viruses have a lipid bilayer which encompasses the protein coat. So in this slide, I have the structure of the virus as well depicted, so that people can refer to what I'm talking about. The envelope viruses are vulnerable to environment like humidity, osmotic pressure, temperatures, pH. Parameters of contact time should be taken into consideration, and their vulnerability to the temperatures. So these are some of the characteristics one has to keep in mind when when you're designing about evaluating the efficacy of the disinfectant. Likewise, for non-envelope viruses which do not contain the lipid bilayer and they just have the genetic material being surrounded by the protein coat, the viruses become much more resistant and they tend to persist in the environment for over a long time, and they sustain over there, and they can sustain in the harsh environments as well. So which includes the extreme pH conditions, the exposure to UV lights, high temperature, as well as the high or low adverse pH conditions. So when we are designing the virucidal efficacy studies, we need to take into consideration that for which kind of viruses are the ones that can be tested on, and do the same disinfectant have a good efficacy against both enveloped and non-enveloped viruses. So this design aspect takes into consideration the type of virucidal agent, its mode of action. When I'm saying the mode of action, does it affect on the protein structure? Does it affects the lipid bilayer, or does it have effect on the genetic material of the virus? Is what one needs to understand correctly, and that's how it helps in the designing of the studies. Then at what concentration or ppms of the disinfectant is effective is one needs to look into. How much of contact time, the conditions in which it has to be tested, is it to be tested in the liquid form, is it to be, and has it to be tested on certain surfaces, or is it being used as an application on the surface, and the applicable surface is what has to be tested by exposing to the viruses. These are the conditions one needs to look into, as well as the temperatures, because sometimes if the temperature is lower, the stability of the virus might be more, as well as the property of disinfectant to have an action may also vary. So those conditions should be looked into. Next is the model viruses that are available to be tested. So when I'm talking about model viruses, it basically means that the viruses should be representative ones that needs to be tested. If the specific viruses are not the ones that can be easily made available, or it may pose a safety risk to the scientist or the laboratory in which the testing might be happening. So the model viruses can be the ones which can represent the categories of viruses that the disinfectant can act on, and hence the guidance documents also clearly tells that the model viruses can be used as the ones representing the category of viruses. So it's very much acceptable as per the regulations. The testing model, do you want to also look into which testing model it will work on? The efficacy can be evaluated correctly. Can it be cell culture based, where you want to have the viruses being infecting the cell system, and the disinfectant and the virus effect can be shown inside the cells, or it has to be, if it is something like a testing of the UV effect on the viruses. So even model like PCR or the molecular method can be the testing model which can be used. So one has to decide that, you can always decide by taking into discussions with the testing laboratory how these designs can be used. Second is, third part is the toxicity. If the test system used is the cell culture based model, does the disinfectant have a cytotoxic effect onto the testing model itself? One needs to overcome that first and then ensure the testing can be successfully performed. Next is neutralization effect. So the disinfectants have, if there is a time point based studies, there has to be neutralization of the effect at those study time points, so that it ensures that the sustained effect can be neutralized and the viruses can be then put into test system for a given time point study. Last is once you overcome the toxicity and the neutralization effect, the test system becomes suitable for testing the virucidal efficacy study. So if one goes into the depth of the guidelines for testing the virucidal efficacy, and the guidance documents clearly says that such studies should be performed in the labs which have the expertise of conducting the virucidal viral studies, and they have the proficiency of handling the viruses, and these should be performed under good laboratory practices. So the good laboratory practices are the regulations that help to ensure that the studies can be, the data coming out has a quality in it, it has reliability, it has traceability, and that's how it adds value to the product. So I'm just going through the guidelines definition of what is GLP. It's a quality system which is concerned with the organizational process and the conditions under which the non-clinical health and environmental safety studies can be planned, performed, monitored, recorded, archived, and reported. So each of these phases is where the good laboratory practices helps to ensure that all the studies can be executed smoothly. They have their data coming out in a reliable manner, and these data can be then accepted by the regulatory agencies in different countries, because the good laboratory practices are encouraged across the different OECD group of countries, as well as in the US and the European Union, and so that the mutual acceptance of the data is practiced across these different nations. And that's how the good laboratory practices being followed helps to ensure that the duplication of these studies in different countries can be avoided. And these good laboratory practices are applied to the non-clinical health and safety studies. So it helps to test pharmaceutical products, pesticides, cosmetics, veterinary drugs, food additives, feed additives, and industrial chemicals. So under in these kinds of categories, the GLP testings can be performed in our laboratories. And the whole purpose of doing the testing under GLP is that the data coming out has quality, and this can be helps to ensure that they are safe for use in humans or in the environment. So now I'm going to talk about the way these studies can be performed under good laboratory practices. So in a typical GLP setup or good laboratory practices being followed in a laboratory, the organizational structure involves that there is a test facility management or overall in charge for a test facility or laboratory who is having the control over the systems in place. He ensures that there are procedures, there are quality systems existing, the equipments are qualified, the personnel have the proficiency of executing the studies, and that's how the test facility management takes the charge and gives the assurance that the laboratory has the right skills and proficiency and the systems in place to execute such studies. So under the test facility management, there are independent QA or the quality assurance team who reviews these kind of studies. There is the test facility has the study directors. It has a TICO, which is test item control office, where they have the facility of accepting the samples from the different customers and ensuring that the confidentiality of these samples and the reliability of data can be ensured. Then there is a document controller, there is an archivist for ensuring that the studies are archived under controlled conditions and they can be made available for the regulatory agencies for during the time of inspections. So under these category, under these different setup of organizational setup in the facility, in the test facility, the GLP studies are conducted. So I will start. I will take a typical example of how the study's flow can be performed, so that it gives an overview for all the viewers that how we can smoothly run through the studies and still have a reliable data coming out, and it helps to fulfill the expectations of the different regulations for that exist across in the different countries, and still be able to give you a quality data out. So it all starts with the customer connecting with the testing laboratory and looking out for a proposal. So first is evaluation that the test facility can perform such kind of studies. So the technical and the scientific discussions take place along with the business development with the customers, that can these studies be really be performed under the existing test facility, and with the agreement between the customer and the technical team, and if the scientific basis of the study can be rightly addressed, that's how it moves into the next phase, where the proposals are finalized and the work orders are issued for conducting such studies in the test laboratory. And thereon, the test facility gets ready for executing these studies. So the first phase involves the shipment of these samples from the customer side to the test facility, and so this forms the part of planning phase of the activities. So it helps to plan the studies for both the customers as well as for the test facility. And thereon, once the sample shipment happens to a test facility, the intimation is given to the test facility management, which helps to ensure that all the sample integrity is there, and basis of that, it intimates to the study directors in the test facility that the studies should be executed, and that's how it is intimated in the form of a study allotment. And basis of the study allotment to the study directors, the study director takes over this part of the study and becomes ready to interact with the customer for preparing a study plan basis of the proposals. So the study plan brings in the scientific design of the study, as well as the compliance aspects into it that would be built in. And these are then discussed with the customers. And once the agreement is there between the customers and the study directors, the study plan gets signed off, which is then which moves into the phase of performing the study, which is the study execution phase, in which the study director gives training to the study personnel under the study plan, and that's how the study plan is. The study personnel gets ready to perform the studies into the laboratories, and this becomes a part of the study execution. But so then it moves on into different phases of the study execution, which I'm going to talk about in my subsequent slides. And it moves into the sample analysis. After sample analysis gets over, all the data gets compiled and the results are tabulated. Then these results are then further analyzed for interpretation of the results, which is then recorded and documented in the reports. So for when we are conducting the GLP studies, it moves into the different phases, and the different departments come into play, and that's what this slide tells about. So it all starts with interaction between the client and the BD. BD is the business development part of the test facility. So where the discussions happen between the BD and the client on the proposal. Once the proposal gets finalized with the techno-commercial discussions, the client starts interacting with the other departments in the test facility, which is the TICO, which stands for Test Item Control Office, where the shipment of the sample is to be done. And the typo department directs the client about the way the sample shipments have to be happened, what all necessary forms or information has to be filled in. And once this information is completely filled in, the samples are shipped, and the test item is received at the typo office, where the samples are encoded so that the confidentiality of the samples is maintained. So here on it, all the details of samples are going to the mode of codes, so that the confidentiality is maintained throughout the period of the study. And these discussions are followed by the intimations to the test facility management. Test facility management then interacts with the study director, and study director is where it is the one who takes over the part of the study, and he becomes the sole in charge till the time the studies are executed and reported back to the client. And that's how you will see in the slide that the study director is the one point contact till the time the studies get over. So here the different departments now come into picture during the phase of planning, performing, executing the studies, analysis, data interpretation, and reporting. So at each phase, the study director is the point of contact between the QA, between the study personnel for the trainings, between the analyst and the QA for the review of the data, as well as recordings of this data, and then documenting this into the report, and then finally archiving the studies so that these studies are made available at the time of regulatory inspections. So continuing my continuing my discussion from the previous slide, the study director is the one who has the maximum role to play during the conduct of the studies, because he's assumed and he's assessed to be fit for on the technical side for taking the decisions on study design as well as execution, as well as his experience comes into play to ensure that the data that is coming out is holds a good quality. So the study director works closely with different departments, liaises with them to ensure that from the time of study allotment, designing the study plan, executing the study, doing the analysis of the samples along with the study personnel, documenting the results, interpreting and reviewing the data, and finally tabulating it and reporting back to the client. The study director has the full control on the conduct of the study. Now I'm going into a little more details of each of these process flow, so that it gives you a better understanding on the phases, how the study moves on. So in the beginning, as I discussed, the request of proposal coming from the client, as well as till the time the work order is issued, there is a discussion or interaction happening between the customer and the testing laboratories technical team as well as the commercial team. And once it reaches to the phase of finalizing the study design, the proposal is finalized and it moves into the phase of agreement and then deciding on the timeline. The next, and then the project moves to the next phase, which is the phase of shipping the samples from the customer side to the testing side and allotting the study. So first phase is the discussion happening between the customer and the test item control office, where the test site is getting ready to receive the samples, but along with the appropriate information on the test item, which is the disinfectant here. And then once all these formalities are completed, the intimation of receipt of the sample translates into intimation to the test facility in charge or management to give an indication to the study director that a testing of a new sample has to be initiated and this is done in the form of allotment of a new study to the study director. So now the interaction has moved from the customer and typo to the test facility management and study director. So in the next phase, the study director gets ready by designing a study plan, and here on the interaction starts for the study director with the customer for finalizing the study design in the study plan, followed by the review of the finalized study plan with the QA, and then the test facility management. So now the discussion starts on the scientific aspects and the compliance part. With the agreement of the study design in the study plan, the customer and the customer signs off on this along with the QA, study director, QA, and the test facility management. The finalized study plan is now ready for execution. So now the project moves from the study plan phase to the execution phase, and here the study director starts interacting with the team of scientific people called as study personnel, so that these study personnel are competent enough for executing the study in the laboratory. So a formal training is what is given to the study personnel for executing such studies. And thereon, during the course of these studies, the study director also liaises with the quality assurance team so that the live inspections can be initiated during the execution phase, so that it gives an assurance that the studies are moving in a compliant manner as what has been agreed upon in the study plan. So the QA inspections happen, which are called as the live inspections or in live phase inspections, and this happens throughout the course of the studies. And for this, some critical phases are the ones that the study director helps the QA to identify, and those critical phases are the ones where the QA inspects the conduct of these studies. So for a disinfectant, during the execution phase, the evaluation of the cytotoxicity is the first phase, where to ensure that if the disinfectant is going to be evaluated on a cell-based system, the cell or the indicator cells are not getting toxic effect, because if that is showing up the toxic effect, the cells will not be able to give the right indications whether the disinfectant is killing the virus or not. So this phase is very essential, and the cytotoxicity has to be neutralized. So the technical understanding of the study director and the study personnel helps to ensure the right design and executing the cytotoxicity studies. Once this is overcome, the study moves to the next phase, which is the interference or the neutralization effect study. So this is the phase where the technical team has to decide now whether this disinfectant is at what stage it will show or it will get neutralized, so that the virus in question is not getting affected by the disinfectant property of the chemical. So the neutralization phase is where this part the study director helps to design and execute this phase. Once these two phases are completed, the contact time of the disinfectant with the model virus and the concentration of the disinfectant is the one which needs to be taken into consideration, so that these two things ensure that in the commercial application, the disinfectant is able to demonstrate this effect on a real-time basis. So these two things come into picture when you are designing the virucidal effects evaluation studies. So after completion of the cytotoxicity and the neutralization effect, the actual virucidal effect evaluation starts off, where the virus in question or the model virus is then tested with a concentration of the disinfectant and the contact time decided for the disinfectant. The virus and the disinfectant is come together, and these ones are then evaluated after those concentrations and contact times onto the indicator cell line to see whether the disinfectant was really able to demonstrate the effect and kill the virus or deactivate the virus. So this was the part of the execution phase of the study. After performing the cytotoxicity, the interference effect or the neutralization effect, and then doing the virucidal evaluation effect, the sample are then further analyzed to see that does the indicator cell really give out those readouts. So these readouts are then recorded and reported by the study personnel to the study director. And on basis of that, the data is then compiled and put it together in the form of results in the report. And this part of data review and interpretation is done by the study director. During this phase, the data interpretation, the study director closely works with the study personnel to ensure that all the data which is coming out from the sample analysis is rightly interpreted and put in the reports. And these are then reviewed by the QA as well to ensure that the raw data coming out from the analysis is correctly being recorded and reported in the reports, so that the results can be rightly attributed to the property of the disinfectant. So after the results are compiled and put it in the form of a report, this report of the draft is then shared with the customer back by the study director, and the study director and the customers and the study director closely work to ensure that all the data is rightly reported out, which is then as well as reviewed by the quality assurance team so as to ensure that all these phases are capturing and the objective of demonstrating the efficacy is rightly put together in the reports. So in my next slide, it tells that during all these phases, the key aspect is the communication that one has to be smoothly going through between the customer and the different departments of the test facility. So you will see that initially the interaction between the customer is with the business development team, followed by discussion with the test item control office or the TICO, and then the customer interacts with the study director, as well as between the study director and the customer. There is also a QA who comes into picture at different phases, so that the reviews are happening, the inspections of the study is rightly happening, and this helps to fill the gap of any of the information that can be that is existing between the customer and the test facility. So in all phases, the communication plays a very key role to ensure a smooth execution of the studies are done. So execution with the conduct of these studies, the advantage is that the GLP system helps to ensure that the data is, the assurance to the data is there, and this helps in the acceptance of these studies by the regulatory agencies in different countries, as well as it helps to fulfill the criteria of mutual acceptance of the data. And the studies can be reproduced at different points of time also, and the data that has come out is traceable and it's reliable, and overall with this it adds value to the product. So now in my last part of this talk, I'm going to talk about how these studies are conducted and Syngene. And this I'm going to tell about in the form of 2 case studies. First, just to give you a briefing of the facility that exists in Syngene for doing this viral efficacy studies is a GLP certified facility. It's a BSL-2 laboratory which is spread over 4,000 square feet. It has well-equipped segregation of activities across different laboratories in form of having a cell culture lab, a virology lab, a PCR lab for doing the molecular studies, and research lab for doing the virus propagation studies. It has also the features of ensuring a biocontainment across different laboratories so that the virus contaminations can be prevented and the aerosols can be contained within the laboratory without exposing to the environment and to the people. It has a dedicated cell bank area for all its analytical cell lines or the indicator testing cell lines. We follow the safe working practices. We have a dedicated suite for conducting the studies. GLP compliant systems and traceability of the data and secure storage is what we assure to the clients. So in the scope of the laboratory does not limit itself to the testing of the products for virucidal efficacy. It also caters to the execution or conducting the viral clearance studies to support the biopharmaceutical molecule programs during its clinical trials and commercial phases. It helps to test the batch release for the bulk harvest of the biopharmaceutical products, as well as testing of the cell banks to de-risk the cell banks from before they are taken into the GMP manufacturing area. So specifically for the antiviral testing, the products are tested based on the cell-based systems. It helps to and the advantages of this testing through cell-based systems is that it helps to quantify the viable viral count, thereby giving the effect that does the virucidal efficacy of the disinfectant can be demonstrated based on the inactivation or killing effect of the compound. And we use model viruses, which helps to ensure that the personnel who are working with these viruses are safe, the laboratory is safe to use these viruses, as well as the model viruses are the representative viruses as per the guideline documents for doing this efficacy studies. And these antiviral studies are completed for the so far for some of the consumer goods companies, for polymer manufacturing companies, and for the surgical gloves. So this antiviral testing, we do it based on some of the cell culture systems that we have in a place, and this is what the slide tells about. So we have the model viruses for the enveloped type as well as for the non-enveloped viruses. And the enveloped and the non-enveloped viruses also falls under 2 categories of genetic material, the DNA and the RNA. We have the appropriate cell systems and test models in place to use those for our efficacy evaluation studies. So now coming to the case studies, I have 2 case studies to depict how we have so far fared in giving out the reliable and the quality data to our customers for the disinfectant evaluations, and one for the polymer evaluation. So here we have used a model virus, which is the parainfluenza 3. The reason being that it has the customer had the requirement that can these virus show similarity to the existing COVID-19 or the SARS-CoV-2 virus, and that's how we chose a model virus which was having an enveloped protein structure and lipid structure, and it had the RNA as the genetic material similar to the SARS-CoV-2, and in terms of size as well and resistance to the physicochemical properties, it was showing similarity to the SARS-CoV-2. So that's how we use the model virus, which was bovine parainfluenza 3, and it gave us an advantage that it was safe to work in the laboratory, as well as it was showing similarity in characteristics to the required SARS-CoV. And the disinfectant was a surface sanitizer that we evaluated, and it was evaluated at a concentration of 1000 ppm, and it showed that with an exposure time of close to 30 minutes, it shows an efficiency of 99.999, ensuring that the entire virus population that was exposed to this disinfectant was deactivated in the span of 30 minutes. And this is just a representative picture of how the indicator cells that were used for testing the disinfectant and the model virus had the effect being demonstrated in the cell system, which helped to interpret the virucidal efficacy evaluation. So a normal monolayer, healthy monolayer is depicted in the first figure, where there's no disinfectant added. In the second picture, the healthy monolayer of the indicator cells was added with the virus to show that when a virus is added, what kind of effect is visible in the cell culture system to interpret the data. As well as in the third picture, what we are showing is that the cell systems, when we added along with the virus and the disinfectant was neutralized to a certain concentration, thereby to neutralize the effect. And this is the concentration that we decide during our neutralization studies and use it for doing the virucidal efficacy studies further. And now I come to the second case study where one of our customers had the polymer with them, and they wanted to use this polymer as a coating onto the hard surfaces, and wanted to understand that when is exposed to these hard surfaces through the contacts, does the virus get inactivated because of the effect of the coated polymer. And again, here we use the same model virus for a contact time of 5 minutes. So the polymer and the virus was kept in contact for 5 minutes, followed by then evaluation onto the indicator cell, and it was surprising to know that within a span of or less than 5 minutes, the polymer demonstrated an virucidal effect of more than 99.99%. And this has further gave confidence to the customer that these polymers can be used on the various surfaces in the market, such as the furnitures or in the aircraft industry or in the transportation industry, basically to coat on those surfaces and to give safety assurance to the customers or the general public. So coming to one of my last slides, the key takeaways from this webinar is that there's a huge demand of the disinfectants in the market, which is projected to grow at a 3% compound growth rate. And testing of these disinfectants in a lab which has the good laboratory practices definitely helps to add value to the product, because it helps to give a quality data, and which adds reliability and reproducibility, thereby giving confidence to the regulatory agencies and to the general public that such disinfectants have a sustained virucidal efficacy, and these data coming out from the laboratories can be reproduced at any given point of time. And during the course of executing such studies, the testing laboratories should take into consideration a good planning, designing of studies by closely working with the client and ensuring a smooth flow of activities, as well as the key aspect for a successful outcome would be a good communication among the different phases of the studies with the customer and internally to ensure that the studies are reliable and they can be demonstrated effectively. So with this, I open the floor for the webinar for a Q&A session.
Oh, thank you, Rahul. That was an awesome session. I've never realized so many things go on in the background of an antiviral product. Given the pandemic and the heightened awareness about coronavirus, it makes so much sense to just understand what goes on before a product hits the shelf and protects us. There are a lot of questions that have come offline and online also. There are quite a few questions that are coming in. So guys, we're a little over the time, but I think given the importance of the topic, we'll keep the questions going on. So if you have a few more, please feel free to drop your questions and we'll pick them. The first one I wanted to pick is from Satya. This question is, why the effectiveness of different vaccine manufacturers are not consistent? What are the factors affecting the outcome?
Sorry, could you repeat the question?
Why the effectiveness of different vaccine manufacturers are not consistent? What are the factors affecting the outcome?
Okay, different vaccine manufacturers, again, one needs to understand that the effect of the vaccine depends on how it interacts once the vaccine is given to the subjects. So some of the vaccines are inactivated kind of vaccines. Some are attenuated forms of vaccines, or nowadays even the mRNA vaccines are there. So it definitely tells that each of these different kinds of vaccines will have a varied interaction with the immune system in the subjects. So, for example, an inactivated vaccine will have only effect till the time it sustains in the body, and to what extent it helps to elicit the immune response in the subjects. Whereas when it comes to attenuated vaccines, these vaccines are live vaccines, so the virus seems to be sustaining there, whereas and it is still active, so that it keeps on proliferating or eliciting the continuous immune response into the body, and that's how those could be effective for a longer period as compared to an inactivated kind of vaccine. So these are some of the factors which come into picture when vaccines are manufactured.
All right. So the next one is from Lakshmi Akshita. Her question is, for testing, what are the positive control used? From where are these procured and stored? And finally, what are the standards that it is traceable to?
Okay, so so I'll give you the example of my testing laboratories. So we procure the controls. We use the controls which are the live viruses. First answer. The second part is we procure these positive controls from the reliable sources like ATCC. So these stocks which are available from ATCCs are also characterized, so that it gives you a reliability that the virus stocks have a good strength, and which is in the form of titer. And the third part of the question is that can you repeat that?
Yes, the third part is, what are the standards that it is traceable to?
Okay, so some of the standards are available or agencies like NIVSC, where the viruses can be procured and they have the known titers coming out from such agencies. Otherwise, if there is if the standard models of these viruses or standard stocks are not available, one can definitely have an in-house stock prepared for these viruses, and they can be characterized and kept in the laboratory to be used as a standard. But they may not be the reference standard, but they will obviously can be considered as an internal standards for performing such studies. And of course, a good test and characterization of these stocks will help you or ensure good, reliable data coming out from the usage of these stocks.
All right, thanks, Rahul. The other one that came live is from Dr. Srinivasa Reddy Yatappu. His question is, Dear Dr. Rahul, if possible, specify the model virus system / pseudovirus system, which can be applicable for screening of antiviral activity of therapeutic molecules.
Mm-hmm. Okay. So antiviral effect is, again, as I said in the beginning of my talk, is that antivirals prohibit the growth of virus and they do not inactivate the virus. So the pseudoviruses, if they have life of only one cycle, it might be a little difficult to design the studies around these pseudoviruses. Whereas if you have, if we have the model viruses which have a similar morphological characteristics to the targeted virus, they can be used for conducting the antiviral studies also. And as I said, it might be even safer for the testing laboratories to use for such viruses during the course of evaluation. So yes, pseudovirus can be used where the study design really permits to perform such studies using those pseudoviruses. Otherwise, you can always bank on two aspects. One is the model virus, which having a similar morphological characteristic, or an attenuated form of the targeted virus, where the infectivity aspect can be nullified and still see the effect coming out.
Okay, so this is a viewer's choice question. The question is, what are your thoughts on antimicrobial resistance, which is on the rise? How can the right effective interventions can be made? So it's another question from Satya.
Okay, good question. There is no one size fits all kind of strategy available here. Each the microbes have their own ways of evolution, and that's how they tend to overcome the effects coming out from the or effects neutralize the antimicrobial properties from using these chemicals. So the multi-pronged approach is something one has to adopt when considering to overcome the antimicrobial resistance. So that's the strategy one has to adopt while taking into consideration that which are the ways which one can overcome this. So for example, if the virus has properties of mutations, with the evolving mutants, one has to keep a track of it and come out with the evolving forms of the antimicrobial products. Otherwise, the same product may somehow become obsolete or become useless after a certain time because the virus might have evolved over it. And that's how one has to keep track of it by doing the evolutionary studies of the viruses. One of the typical example is like how the influenza vaccines are manufactured every year, looking at the changes in the mutants that are evolving in the environment. Similar approach one has to adopt in this case.
All right, thanks, Rahul. I'll pick up two questions that came in offline. With that, we'll wrap up the session.
Okay.
The first one is sort of closer to what Kivani does, in the sense that does all virucidal efficacy studies require a BSL certified lab?
So when we are working with viruses, definitely you need to consider that the biosafety is something one has to keep in mind, so that it protects the environment and the people and the scientists who are working with such viruses during the time of studies. However, if there is a model system which is which helps to reduce the risk, like the pseudoviruses, still it has to be conducted under the a lower risk category of biosafety, but yes, essentially under a biosafety conditions. So whether it's biosafety level 1, 2, 3, or 4, the requirement of biosafety would be there while conducting such studies.
Thank you. And the final question for you, before we let you go, what about herbal and Ayurvedic antiviral products? Do they also need to go for the studies?
The studies will definitely help to demonstrate the spectrum of the viruses that the herbal or the natural products have capacity to inactivate, because only after testing them we get to know which all viruses the products are effective against. So one such example is we happen to test the extract of garlic on the envelope viruses, and we found it to be quite effective. Till that time, we were not so much aware that they are really helpful in our day-to-day life in combating the viruses. So yes, a study of the herbal products or natural products will definitely help to give you a more understanding in their efficacy.
All right. So on the note of garlic, let's bring this wonderful session to a close, Rahul. Rahul, thank you so much. I mean, it's been an eye-opening session for me. I'm sure that's how the viewers also find it. So there are a few unanswered questions. We will post the responses to it offline on our website.
Yeah, you can share those questions. I would be happy to take them up offline and respond back on them.
Yeah, wonderful. Thank you. Along with that, a copy of the presentation that you used and also the recording of this webinar will be published. So, just follow us on our social media channels and you will get to know when we are publishing it. So thank you so much for joining. Have a good weekend wherever you have joined in from. And so this is Perspectives, a thought leader initiative by Kivani. Rahul, thank you. And thank you everyone.
Thank you.
Bye-bye.
Bye-bye.